The overall goal of this procedure is to systematically tease apart how different types of social inputs differentially relate to infant neural activation by recording infant EEG in both social and non-social contexts.
The main advantage of this technique is the inclusion of control social and non-social conditions to examine effects on infant EEG of different social inputs, including language and face-to-face interaction. This method has implications for the field of developmental cognitive neuroscience, highlighting the need to consider the role of EEG recording context when assessing functional neural activation.
This method can be applied to the study of individual differences to see how EEG activity in the presented conditions varies by factors such as socioeconomic status, age, or parenting quality.
We first had the idea for this paradigm when we noticed that many studies have recorded infant EEG during baselines that combine both social and non-social elements.
It is best to conduct this experiment in an electrically shielded booth to prevent interference with the EEG signal. Position 2 speakers on the ground on either side of the table directed towards the chair. Place a tripod video camera under the table and facing up so that the infant's face will be in clear view during the experiment. Hang an opaque curtain immediately behind the table so that the experimenter can stand behind it to be hidden from the infant's view during parts of the paradigm. Finally, place cereal, a small stuffed animal, and a rattle in the booth. Obtain informed parental consent and explain the EEG experiment.